Hello, I'm Dr. James Billis. We're going to cover Chapter 19, The Blood, out of the textbook Fundamentals of Anatomy and Physiology, 11th edition. The blood and the cardiovascular system. The cardiovascular system consists of a pump, which is a four-chambered organ, muscle, a series of conducting hoses, the blood vessels. We like to look at the blood vessels as being arteries and veins, where the arteries are the color red as they carry oxygenated blood, and the veins are colored blue as they carry deoxygenated blood. And this will work until we get to the pulmonary system, and we'll have to change it around a little bit. But for now, we'll look at it in those terms. The components and functions of blood. Blood is a specialized connective tissue. It contains cells suspended in a fluid matrix. Functions of blood are to transport dissolved gases, nutrients, hormones, and metabolic wastes, regulating pH, ion composition, and interstitial fluid, restricting fluid loss from injury sites, defending against toxins and pathogens, stabilizing body temperature. And we'll discuss all these more in detail as we move on. Components and functions of blood. Some of the characteristics of blood, temperature-wise, 100.4 degrees Fahrenheit. It has high viscosity, which is greater than water. Slightly alkaline pH, which is 7.35 to 7.45. Blood volume, 7% of the body weight. Therefore, a 165-pound person would have approximately 5.4 quarts of blood. Components and functions of blood. Whole blood. When we refer to whole blood, we're talking about if we were to do a blood draw from a superficial vein in a person, what we would have in the tube would be whole blood, which consists of both plasma and formed elements. And the idea, what we're going to try to do is we're going to try to separate that plasma from the formed elements. And when you separate that, it's called fractionation. It's a process of separating whole blood into plasma and formed elements. We will do this using a process called a hematocrit. Hematocrit will take a capillary tube and put some blood in the capillary tube and spin it down in a centrifuge. And when we spin it down, the heavier products inside the whole blood go to the bottom, and we end up with a red layer, and on top of that red layer, a clear layer. The red layer is typically the formed elements, and the clear layer is the plasma. Now, plasma makes up about 55% of the blood volume. More than 90% of the plasma is water. Okay, so a majority of the blood is plasma, and 90% of that is water. It contains dissolved plasma proteins and other solutes. The most abundant protein in plasma is albumin. Plasma is similar in composition to interstitial fluid. Not water, but interstitial fluid, because water ions and small solutes are exchanged across the capillary walls. Let's see, what we're looking at here is we discussed the hematocrit a little bit. The hematocrit is the percentage of formed elements in a sample of blood. The normal hematocrit or packed cell volume in an adult male is 46, whereas with a female, it's 42. The sex difference in hematocrit primarily reflects the fact that androgens, which are male hormones, stimulate red blood cells production, whereas estrogen in the female hormones does not. As we look at plasma and formed elements here, it's saying, again, 55% of the spun down blood would be plasma, 45% would be the formed elements. Within the formed elements, what we've separated out is red blood cells. 99.9% of the formed elements is red blood cell, white blood cells, and platelets. In plasma, again, 55%, Most of it is water at 92%, other solutes, and then we have some plasma proteins as well. Components and functions of blood. We talked about plasma proteins. Plasma proteins, again, albumin is at 60%, the most abundant protein in the plasma. And then we have globulins at 35%. And fibrinogen at 4%. Soluble protein that functions in clotting. And in addition, we have other proteins with varying concentration of enzymes and hormones. Discussing origins of plasma proteins, more than 90% are made in the liver, including all the albumins, fibrinogen, globulins, and various proenzymes. Antibodies are made by plasma cells. Peptide hormones are made by endocrine organs. We've touched on this a bit. The components and functions of blood. We have formed elements, which consist of red blood cells, white blood cells, and cell fragments, which are platelets, which aid in clotting mechanisms. Hemopoiesis is the process of producing formed elements. Another term for hemopoiesis could be hematopoiesis, and the hormone that we're referencing through the process of making red blood cells is hematopoietin, which we make the red blood cells in the bone marrow of the bones. In discussing platelets, they are small, membrane-bound cell fragments that contain enzymes and other substances important to clotting. White blood cells, there's several different types of white blood cells, which we'll get into. We call them leukocytes. Leuko meaning white, eryth, just a little more background for you, a review. Eryth being red, xantho being yellow, cyano being blue. Again, leuko, white, and it lists the different types of white blood cells, and we'll get into those a little further as we move on. Red blood cells, erythrocytes, eryth being red, are the most abundant blood cells. Again, we saw that was 99.9% of the formed elements. These specialized cells are essential for the transport of oxygen in the blood. Red blood cells, also called erythrocytes, make up 99.9% of formed elements, contain hemoglobin. Hemoglobin provides a red pigment that gives whole blood its color, binds and transports oxygen and carbon dioxide. A red blood cell count. The number of red blood cells per microliter of whole blood in the adult male is 4.5 to 6.3 million. In the female, it's 4.2 to 5.5 million. Hematocrit, we've discussed, again, is packed cell volume, the percentage of formed elements in the blood. In the adult male, it's 46. In the female, it's 42. And again, the difference between those two numbers are going to be with the male, the androgen, versus the female, the estrogen. Red blood cell, the structure of the red blood cell. It's small, highly specialized cell, biconcave discs. Biconcave discs. A thin central region and a thicker outer margin. Oops, I lost it. Let's go back. When looking at a standard blood smear, the red blood cells appear to be two-dimensional objects because they are flattened against the surface of the slide. This is not how they actually are. This picture here represents a 3D version, and you can see that they're not flattened. Important to note, as these red blood cells work their way down to the smallest vessel that we've indicated as far as arteries and veins, the smallest one is a capillary. And as these red blood cells pass through the capillary, The capillary is so small that only one red blood cell at a time can pass through the capillary. This is showing you the size and the shape, concaved, larger on the outer portion, thinner on the middle portion. Important effects of red blood cells, structure on function. Let's see, we've got a large surface area to volume ratio. They quickly absorb and release oxygen. They form stacks called, a French word called rouleau. Smooth blood flow through narrow blood vessels. They bend and flex when entering the small capillaries. As I indicated, only one can pass through at a time. But as you can see, a small capillary can, or a larger red blood cell can pass through a smaller capillary because it will flex and bend. What we're seeing here is a picture of these capillaries. And within these capillaries, we can see individual red blood cells being stacked. We call that, again, stacked red blood cells, rouleau. Now, the mature red blood cell is anucleate. It lacks a nuclei. It has no mitochondria or ribosomes. It is unable to divide, synthesize proteins, or repair damage. Important, the mature red blood cell lives about 120 days or 4 months. Hemoglobin is a protein in the red blood cell that transports respiratory gases. The hemoglobin consists of 4 globular protein subunits, those being 2 alpha chains and 2 beta chains, each with 1 molecule of heme. Each heme contains 1 ion. The iron interacts with the oxygen to form oxyhemoglobin, dissociate easily to form deoxyhemoglobin. In this picture, we're looking at the sectioned red blood cell, and we see an alpha chain here, an alpha chain 2 here, beta chain 1 here, beta chain 2 here, and we can see a heme group, 1, 2, 3, 4 heme groups. And in the heme group, what we're looking at is lots of carbon, hydrogen, oxygen, and in the center of the heme group, we've got iron. 4 of those heme groups within a molecule of hemoglobin. When we discuss fetal hemoglobin, there's a form of hemoglobin in embryo or the fetus, binds oxygen more readily than does adult hemoglobin, takes up oxygen from maternal blood at the placenta. Hemoglobin function. Each red blood cell contains about 280 million hemoglobin molecules. Each red blood cell can carry over 1 billion molecules of O2. In peripheral capillaries, where O2 is low, hemoglobin releases O2, binds CO2, forming carbaminohemoglobin. At the lungs, where O2 is high, hemoglobin binds O2 and releases CO2. Oxygen and carbon dioxide. Anemia. This results when the hematocrit content of red blood cells is reduced. Interferes with oxygen delivery to peripheral tissues. Red blood cell formation and turnover. 1% of circulating red blood cells are replaced per day. About 3 million new red blood cells enter the bloodstream each second. The term erythropoiesis, red blood cell formation. Eryth being red, poiesis being formation. In embryos, embryonic blood cells move from the bloodstream to the liver, spleen, thymus, bone marrow. They differentiate into stem cells that divide to produce blood cells. Now that's in the embryo. In adults, the process of erythropoiesis occurs only in myeloid tissue found in red bone marrow. Hemocytoblasts, also called hematopoietic stem cells in the myeloid tissue that divide to produce myeloid stem cells, which become red blood cells and some white blood cells, and lymphoid stem cells, which become lymphocytes. Hematologists, or blood specialists, have identified several stages of red blood cell maturation: myeloid stem cell to proerythroblast to erythroblast stages to reticulocytes to mature red blood cells. This picture shows you the formation of the red blood cell from the red bone marrow. Day 1, we have the proerythroblast, changing to the erythroblast. Day 2, basophilic and erythroblast. To day 3, polychromatophilic erythroblast. To day 4, normoblast, the ejection of the nucleus, leaving the reticulocyte, enters the bloodstream as a mature red blood cell. Okay, we talked about erythropoiesis, the formation of red blood cells. Now we're going to talk about erythropoietin, which is the hormone that stimulates erythropoiesis. It's secreted by the kidneys and liver when oxygen in the peripheral tissues is low. We call that hypoxia. When oxygen in peripheral tissues is low, hypoxia. Blood doping, a dangerous practice used by athletes. Reinfuse packed red blood cells to elevate hematocrit. Ultimately, what they're after is more oxygen in the system so the muscles can perform at a higher level. A lot of athletes will train in higher altitudes and then compete in the lower altitudes. Erythropoiesis requires amino acids, iron, folic acid, and vitamin B12 and B6. Lack of B12 leads to pernicious anemia. Hemoglobin recycling. Macrophages, the eaters, cleaners, macrophages of the spleen, liver, and red blood or red bone marrow engulf aged red blood cells. Again, we said they live 120 days. They could be damaged too. Remove hemoglobin molecules from hemolyzed or ruptured red blood cells. Break the hemoglobin into components, and only the iron of the heme unit is recycled. Hemoglobin recycling, we've got a term, hemoglobinuria, the red or brown urine due to abnormally high hemolysis in the bloodstream. Hematuria, whole red blood cells in the urine due to kidney or blood vessel damage. Hemoglobin recycling. Iron is removed from each heme unit, as indicated before, forming green biliverdin. It's converted to an orangey yellow bilirubin. The bilirubin is excreted by the liver in bile. Jaundice is caused by a buildup of bilirubin. When iron is removed from the heme, it is bound and stored in a phagocytic cell or released into the bloodstream. In the bloodstream, iron is bound to transferrin. Developing red blood cells and red bone marrow absorb transferrins and use them to synthesize hemoglobin. Excess transferrins are removed in liver and spleen, storing iron in ferritin and hemosiderin. Blood types. Surface antigen, substances on plasma membranes that identify cells to the immune system. Normal cells are ignored and foreign cells are attacked. Blood type, determined by the presence or absence of surface antigens on the red blood cells, thus leaving us to A or B or RH being D. I'm sorry, let me clarify that. the blood type determined by the presence or absence of surface antigens on the red blood cell, those antigens being A, B, Rh, or D. There are 4 blood types. Type A, which has a surface antigen A. Type B has a surface antigen B. Type AB has antigens A and B. Type O, neither A nor B antigens. Rh blood group is based upon the presence or absence of Rh antigen. So a positive Rh surface antigen is present. An example would be type O positive. It's showing positive for the antigen. Whereas the antigen, Rh antigen, is negative, our example would be type O negative. The antigen is not there, it's absent, therefore it's negative. The term agglutinogens is surface antigen on the red blood cell, screened by the immune system. Agglutinins are the antibodies in plasma, and they attack antigens of foreign red blood cells, causing agglutination or clumping of foreign cells. Agglutinins type A, you would see anti-B antibodies. Type B blood, you would see anti-A antibodies. Type O blood, both anti-A and anti-B antibodies. Type AB blood, neither anti-A nor anti-B antibodies. Type A blood has red blood cells with surface antigen A only. And one thing to note, it's based on shape. Your antigen surface is based on shape. If you have type A blood, your plasma contains anti-B antibodies, which will attack type B surface antigens. If you have type B blood, your red blood cells has the B antigen, surface antigen. If you have type B blood, your plasma contains anti-A antibodies, which will attack type A surface antigens. Type AB blood has red blood cells with both A and B surface antigens. If you have type AB blood, your plasma has neither anti-A nor anti-B antibodies. If you have type O blood, your red blood cells lacking both A and B surface antigens. If you have a type O blood, your plasma contains both anti-A and anti-B antibodies. Again, your blood type depends on the presence of surface antigens called agglutinogens on the red blood cell surface. The plasma contains antibodies, agglutinins, that will react with foreign surface antigens. Cross-reaction or transfusion reaction may occur in a transfusion of blood or plasma from one person to another. Occurs if the donor and recipient blood types are not compatible. Plasma antibody meets its specific surface antigen. Red blood cells agglutinate and may hemolyze. The following picture is a cross-section of antibodies which react with target antigens, causing agglutination and hemolysis of the affected red blood cells. In this example, anti-B antibodies encounter B surface antigens, which cause the red blood cells bearing the B surface antigen to clump together and break apart. Compatibility testing is performed in advance of transfusions. Cross-match testing reveals cross-reactions between donors' red blood cells and recipients' plasma. Type O negative is the universal donor, but cross-reactions can still occur as at least 48 surface antigens exist besides A and B. This picture is showing us the various types of reactions. When we look at anti-A, we're saying that the agglutinin or the antigen has I'm sorry for the pause. My dogs Bella and Sugar Bear were fixing to go crazy as a visitor came to the door. So getting back to this, we have the antigen, the A antigen and the A antibody attacking one another, creating clumping. There's no attacking going on as far as the B, and the anti or the RH did attack. Therefore, we have positive here, and we have a positive here. So we call this a positive blood type. When we look at this one here, we see that there's clumping on the B. So the B antigen and the B antibody have attacked each other. Nothing here on the A. And yes, again, we've got clumping here on the RH. So what we have is a B positive. Positive or negative is making reference to the RH, clumping or not. Here we have clumping, the antibody and the antigens have attacked one another on the anti-A, and they've attacked each other on the B, and there is clumping. So we have AB positive. No clumping here, no clumping here, and no clumping here. So this is neither A, neither B, or neither RH. So we have O negative. There is a problem called hemolytic disease of a newborn. We don't want our RH antibodies attacking antigens, RH antigens, but sometimes this happens when a woman gets pregnant, and problems seldom develop during a first pregnancy because very few fetal cells enter the maternal bloodstream. Thus, the mother's immune system is not stimulated to produce anti-RH antibodies. The most common form of hemolytic disease of a newborn develops after an RH negative woman has carried an RH positive fetus. Exposure to fetal red blood cell antigens generally occurs during delivery when bleeding takes place at the placenta and uterus. Such mixing of fetal and maternal blood can stimulate the mother's immune system to produce anti-RH antibodies, leading to sensitization. About 20% of the RH negative mothers who carried RH positive children become sensitized within 6 months of delivery. Because the anti-RH antibodies are not produced in significant amounts until after delivery, a woman's first infant is not affected. In a future pregnancy, if future pregnancy involves an RH positive fetus, maternal anti-RH antibodies produced after the first delivery cross the placenta and enter the fetal bloodstream. These antigens destroy fetal red blood cells, producing a dangerous anemia. White blood cells, WBCs, are called leukocytes, have nuclei and other organelles. They lack hemoglobin. Their functions are defending the body against pathogens, removing toxins and wastes, attacking abnormal and damaged cells. White blood cells can be found in connective tissue proper, organs of the lymphatic system. Small fraction of white blood cells circulate in blood, 5,000 to 10,000 per microliter. Types of white blood cells are the neutrophils, eosinophils, basophils, monocytes, and lymphocytes. Here we see a picture of a neutrophil, an eosinophil, a basophil, a monocyte, and a lymphocyte. Some characteristics that you might be able to pull out here is we have 3-lobed nuclei in the neutrophil. which is the most abundant white blood cell in the body. Here we have an eosinophil, which is stained orange. Again, it's got a multi-lobed nuclei. You'll see this associated with parasites. We see, in this one, we see a basophil, which is granular in nature. We see a large monocyte here. The monocyte appears to be very large in comparison. And here we see a lymphocyte. So different types of white blood cells. The neutrophil, also called the polymorphonuclear leukocyte, that's a mouthful, 50 to 70% of circulating white blood cells, most abundant. Pale cytoplasmic granules, so it is granular, containing lysosomal enzymes, bactericidal, bacteria-killing compounds. So the way to remember this is neutrophil associate with bacteria killing. They are very active phagocytic cells which attack and digest bacteria. Eosinophils, another term is acidophils, 2 to 4% of circulating white blood cells, engulf bacteria, protozoa, and cellular debris, attack large parasites by releasing toxic compounds, sensitive to allergens, release enzymes that reduce inflammation caused by mast cells. Eosinophils stain red, which is different than the other white blood cells. And you think eosinophils, think parasites. They attack large parasites. And here's a picture of your eosinophil. Basophils, less than 1% in circulating white blood cells. Cross capillary endothelium and accumulate in damaged tissues. They release histamine and heparin. Histamine dilates blood vessels, heparin prevents blood clotting. And here's a picture of a basophil. Notice it's granulated, has nucleus, and is large. But in comparison, the same approximate size as the neutrophil and the eosinophil. The monocyte, which is the largest, 2 to 8% in circulating white blood cells, remain in the bloodstream for 24 hours. Then enter the peripheral tissues to become macrophages. They're aggressive,